Nothisha abanethulela izifundo zika grade 12 zonke izinsuku kusukela ngoMsombuluko kuze kube yiSonto. Zithokozeleni lezi zifundo, nisithumelele imibuzo lapho nidinga khona usizo.
Sanibonani bafundi baka grade 12. Lona okhulumayo uthisha uMr BS Mabaso ofundisa ePhendukane Full Service High School, ofundisa i-Life Sciences. Sizoqhubeka lapha egcine khona nge-genetics. Siqhubeka nje ngama different topics, subtopics genetics. Namhlanje sizobheka i-monohybrid cross kanye nama types of dominance, monohybrid cross and the types of dominance. Lapho nje ake sikhumbuze ama terms asebenza kakhulu when you talk of genetics: heterozygous and a homozygous. Lapho sithi heterozygous ilapho ama alleles engafani, kanti homozygous ilapho ama alleles efana. So no dominance no recessive. Uma sithi gene dominant should ilona elibonakalayo einganeni elivala lelinye igene eli recessive elingabonakali kodwa likhona. Lokho kusho ukuthi na uma ubheka umehluko phakathi kwe-phenotype ne-genotype. I-phenotype is the physical appearance, ilokho ukuthi uma ubona enganeni ubona ngamehlo, alright, kuma offsprings, alright. Kanti i-genotype you cannot see it with our naked eyes, but it is there. Like for instance, uma uhlanganisa u-tall no short kuvele ingane ende, phenotypically leyo ngane inde siyayibona. But masubheka i-genotype yayo inalo i-gene lobufishane. Then la ku-monohybrid cross uzobheka ufuzo olulodwa. One mono means one trait or one characteristic. Before we look at that one, let's go back a little bit and look at our experiments ka-Greg Mendel. uGreg Mendel kuthiwa father of genetics ngoba uyena uqala umuntu owabheka ufuzo kanye nokuhlukahlukana ngofuzo uma siwa kuzizukulwane esilandelayo noma kusuka kuzizukulwane esinye kuya kwesinye from generation to generation. And uGreg Mendel wayengazi lutho ngo-DNA, engazi lutho ngo-chromosome, but he looked at the physical features. Ucwaningo lakho lokuqala wasebenzisa izihlahla zophizi noma izitshalo zophizi. Wathatha isitshalo eside nesitshalo esifishane, alright, waseyihlanganisa. Uthe maseyihlanganisile mayebheka ingane zakhona zonke izingane zazizinde, wasekhanda-ke lo mthetho esithi i-principle of dominance. Lapho sibona ukuthi ama-offsprings akhombisa noma izingane zikhombisa i-phenotype noma ubunjalo bomzali oyedwa. Yiza ehlukene umuntu nomfishane ukuthi ingane zonke zinde. So the principle one gene mask the other gene. That is the principle of dominance. All right? And then obviously the ingane ke manje zinde ezayibizwa nge-F1 generation wayibiza nge-F1 generation. Abazali wabiza nge-P1. U-P1 umele u-first parents, abazali bokuqala. Then iy'ngane zabo wazibiza ngo-F1. F1 umele u-first filial generation, izingane zokuqala. Wabe sethe le ingane zokuqala ke zinde wayihlanganisa. Uthe mase ethola imphumela yakhe wathola ukuthi sekukhona nezingane ezifishane. Le ngane ezinde zombili zazala izingane ezinde ezingathi 75% eyintathu, mase one characteristic eyintathu bezizinde, eyodwa yasibafishane. Wase yabona ukuthi okay, lapha uma kwenze kudlula ufuzo ayahlukana ama genes, ayithi nuclein ngesikhathi leso ayahlukana. Wase from a principle of segregations. Ithi genes separate during the process of meiosis. All right. Masibheka ke le monohybrid cross, into esoyibheka kakhulu is the principle of dominance. So sebenzisa a principle of dominance. All right? Siyawasolva ama-genetic problems. Lesiqale ngayo le experiment recommended. Ukurecommended kuthiwe microsa lapha utall no short. And utall is dominant. So uma uyibhala lapha uyayisa gene dominant. Tall is dominant over short. All right? And then wasebhala waseyenza lapha abazali bokuqala P1 wenza iphenotype yakhona. Lapho kuhlala ushort, sorry, yes, utall short nobani no tall. Iy'tshalo ezimfishane netshalo ezinde. That is P1. So tall crossed with short. Phenotype le. And you get a mark for that. Thole mark lapho. Ukubasho nje abazali. Osukunikeziwe bona. So uyabothi imali lamahhala lelo. Ngoba uma bekunikeza i-genetic problem, bayakunikeza i-phenotype. Nokuthi abazali banjani. All right? One mark lo. Wesibili, suyazi ukuthi abazali they are either homozygous or heterozygous. So P1 kwathiwa he crossed the homozygous tall pea plant and a homozygous short pea plant, which means that i-genotype yakhona izoba ama-capital letter T because tall is dominant and small letter T for short. Therefore, genotype will be homozygous tall crossed with homozygous short. Uthola futhi elinye imaki lamahhala lapho. All right? Sindlule lapho sokuzoba i-process of meiosis. Lapho sekhandeka manje ama-gametes la esikhulume ngawo in our first lessons la awobulili. Ama-male gametes azoba lama genes ka-tall, ama-alleles ka-tall. And remember, ama-genes always occur in pairs. Nangapha futhi is occur in pairs. Then after meiosis, each gamete will have one gene ka-tall. Nangapha futhi each gamete is going to have one gene ka-short. Bese kwenzeka i-fertilization. Remember, asamsebenzisi fusion, sisebenzisa fertilization. So mfundi noma uthola incwadi uyifunda uthola igama lithi fusion lapha. Uma uzohlala ama-gametes, please cancel it and write fertilization. Kusho ngani lokho na? Because masikhuluma ngo-fusion sizokuhlala nama-72 kanti lana i-nucleus of the sperm ihlangana ne-nucleus of the ovum. Bese kuphuma a diploid zygote. So lapha ke sekuvela manje iy'ngane ke ezinde zonke. And ley'ngane uma uyibheka i-genotype yakhona inoba na ino tall no short. But u-short akabonakali, kubonakala bana u-tall in their phenotype. That is the principle of dominance. And bese uyambhala ke u-F1 generation. You get another mark for that one. So you get a mark for ukubhala P1 no F1 imaki lelo lamahhala. Nokubhala umeiosis kanye noba na no fertilization. This is another mark. So each genetic problem all in all iba no 6 marks. All right? And kulo 6 marks lo 4 marks simbiza ukuthi ama low hanging marks. Imaki le phenotype and the genotype and the meiosis no fertilization kanye no P1 no F1. All right? All right, I guess bheke manje ke ama types of dominance ngoba sikhulumile indaba ye principle of dominance. So the first one is the complete dominance. So la complete dominance na it refers to a genetic cross where a dominant allele masks, all right, or blocks the expression of a recessive allele in a heterozygous condition. I-dominant la kusho ukuthi na leli gene elivelayo ezinganeni limboza leli gene elingaveli. That is the principle of dominance. All right. And then lapho complete dominance senza njani na? How do we go about solving a genetic problem, your complete dominance? We follow the steps. The first thing, there is a genetic problem. 1. In humans, ability to roll the tongue is due to a dominant allele. And a man who is heterozygous for tongue rolling and a woman who cannot roll her tongue have children. Use the symbols T, capital letter T, and small letter t for the alleles of the tongue rolling characteristic and represent a genetic cross to determine the possible genotypes of and phenotypes of their children. 6 marks loyo. Bathi nana ugoqa ulimi. Khona abantu abakwazi ugoqa ulimi, khona abakwazi ukugoqa ulimi. Sekuthathana manje ke ubaba nomama. Ubaba kuthiwa heterozygous for tongue rolling. All right? And umama is a non-tongue roller. And tongue rolling is dominant over non-tongue roller. And also baphinde bakunikeza futhi ama letters ufuna usebenzisa na uletter T. And mfundi ke lapha ke sonke isikhathi mawuza genetic problem mabe kakunikezanga i-letter oyisebenzisayo eye-dominant gene. Alright? Now for example, a red flower is crossed with a white flower. Red is dominant over white. So lapho ngingakuthi red dominant, we're going to use R as a capital letter for a dominant gene and small letter r for a recessive gene. Alright? And then lapho kubalulekile you must first read the problem carefully. Best into yokuqala is the phenotypes of the men and the women. So emoqala nje the problem yakho ke lasikahle bese ngiphinde kwesibili manje ubhala uP1 kulo P1 loyo abazali la and then you write their phenotype kubabuye kanjalo. So lapha bathi ubaba is heterozygous tongue roller. So phenotypically lo baba is a tongue roller. So we have to apply for first parent, a tongue roller, and then ubaba, but umama cannot roll the tongue, which means she is a non-tongue roller. So I'm going to use bana non-tongue roller. That is their phenotype. That is their phenotype. So P1 phenotype, and most actually get tongue roller and crossed with non-tongue roller. Second step, you write the genotypes. This palamanja ama genotypes. Baba is heterozygous. And remember, if it is heterozygous, it means that the genes are different. So I've got a gene for tallness and a gene for shortness. So Baba, you're going to write capital letter T and small letter t. And you know that so this alleles go in pairs. Mama is homozygous for non-tongue rolling. So umama, we're going to write or small letter t ababili. Right? And remember, when starting out, you know that a hybrid, baba is a hybrid. So hybrid is a heterozygous. Umama is a pure breed. A pure breed is a homozygous. Alright? That is P1. So if I identify the genotypes, you get a mark for that. The next step is the step of meiosis. Alright? This is our step 3. So we know that during meiosis, the number of chromosomes is reduced to half. Okay? And 2, the cell divides into 4 cells, which are called gametes. So the Bama is a cells obulili, 2 gametes or sex cells. Kubaba, sperm, kumama, amaqanda. Alright? Eggs and the sperm. Abana, they are haploid. So which means that ubaba uzokhipha ama-sperms are haploid. Ka short nama-sperms abani ka tall. So uyabhala lokho bese uyasusa lapha ke kwenzeke i-principle of segregation. Uyaseparator u-tall uzohamba yedwa, u-tongue rolling uhamba yedwa, no-non-tongue rolling ahambe yedwa. Capital letter T and small letter t. So after you have written the gametes, you write meiosis phezulu, ngenzansi ke bese you write down the gametes. You write gametes lapha ekuqaleni. And then you write the gametes. Ubaba uzoba nama sperm ka tongue rolling, nama sperm gani ka non-tongue rolling. Whereas umama is homozygous for non-tongue rolling. So umama uzonoba na small letter t bodwa. all right? And you get the mark for that. After that, there was any fertilization. You write fertilization. And then next, tongue rolling kababa no non-tongue rolling kamama ukhipha ingane uba na tongue roller, but heterozygous for tongue rolling. Nomunye futhi tongue rolling non-tongue rolling lo kababa manje mase hlangana no non-tongue rolling kabani kamama sekuvela ingane na non-tongue roller. So mawubheka lapho ke ezimbili zezo ingane lezo so ama-tongue roller ezimbili zingama non-tongue roller. So ukubhala i-genotype leyo ukuthi ke 2 non-tongue roller and tongue rollers imag lelo, okay? Imag the genotype and their phenotype. And don't forget to write F1, first filial generation lezo ingane lezo, all right? Then masihlanu kama marks kuba about 7 to 8, okay? But still 6 marks, all right? And remember, any mark allocation you can go P1 and F1 imag. Fertilization and meiosis is another mark. So we've got 4 low-hanging marks. So no matter what data is in the problem that solve, you can get 4 out of 6. Remember that in an examination. So I'm going to have food mark being as a problem. And I attempt. So we must have this format again. P1, F1, phenotype, and genotype. All those 4 things are given in your genetic problem. But you have heterozygous, so ubaba uzoba no capital letter T no small letter T. Mama is homozygous because it's a non-tongue roller and she has a recessive gene. You use the small letter T. And that principle is called complete dominance. All right? And then the second type of dominance is the incomplete dominance. Incomplete dominance. Ngamanye amazwi lapha ke alikho igene elistrong kunelinye. So makuhlangana igene kababa nelika mama kuvela into ephakathi nendawo. Like for example kuhlanganisa isitshalo esineflower elimhlophe neflower elibomvu. Omabili aba dominant. All right? And then mayise hlangana lapha azokhipha into entsha isitshalo esinamaflower apink. That is called incomplete dominance. It refers to a genetic cross between 2 phenotypically different parents, producing an offspring different from both parents, but with an intermediate phenotype. Okay? So, 2 contrasting parents, a plant with a red flower and a white flower. But the offspring will have the intermediate characteristic, which is not the same as the mother and the father. Or red no white, or pink. And that is called incomplete dominance. All right. Let's look at the example then, a genetic problem 2 now. What actually happens there. All right. A homozygous snapdragon plant with red flowers was cross-pollinated with a homozygous snapdragon plant with white flowers. All right. And for our symbols, we don't use capital letter and small letter. We use capital letters for both. So it will be capital letter R for red and capital letter W for white flowers. All right. All the plants that grew from this cross had pink flowers. All right. Then there is the question. Represent the genetic cross to show the possible genotypes and phenotypes of the F1 generation in plants. So we follow the same procedure. We write P1, the phenotype is red plant, and the genotype is white plant. So the red plant crossed with white plant, it gives you a mark. Then you write the genotype, homozygous red and homozygous white. And Lapo, remember to use the capital letters because it is the principle of dominance. So there would be capital letter R and capital letter W. Then usukipa amagametes through in meiosis. So the red flower is going to produce gametes for red, and the white flower is going to produce gametes for white, and they're both written in capital letters because it is the principle of dominance. All right, and then we have fertilization. So when you cross a red flower and a white flower, you get a genotype, which is capital letter R and capital letter W. And the phenotype, all the plants will be pink. And that is your F1 generation. All right, asiqhubeke sibheke futhi yona incomplete dominance now. Lana incomplete dominance now sesithatha manje F1 lama flower pink and cross with another F1. Ngoba sesicross iflower eli pink nelinye iflower eli pink. All right, F1 cross by bana F1. Masuyibhala lena uthi P2. These are the second parents. Phenotype, a pink flower is crossed with another pink flower. Sihlanza iflower eli pink nelinye pink. You get a mark for that. Genotype yakhona uzokuba na. Genotype yakhona will be capital letter R and capital letter W crossed with another capital letter R and capital letter W. Then that is followed by meiosis. Then ngapha kumale uzokuba na a sperm for red and a sperm for white. Same thing ngapha ku female. We're going to have an egg for red and an egg for white. All right. Then most cause all red, no red. Say we are red lawyer or homozygous red. Sekuvela iflower elibomvu. Okay? Ubana ongabazali noma kugogo or umkhulu. All right? Most cause no red, nalo white sekuvela ini iflower elipink. Mopina futhi ukrossa white wangapha manje kumale no red wangale ku female. Vela iflower na eli pink. And then ekugcineni usukrossa white wangapha kumale no white wangale ku female. Sekuvela iflower elinjani na eli white. Uyabona usiyabuya lo white lo. Sesibuya ki principle na, principle of segregation. All right? And then if phenotype is na 1 red, 2 pink and 1 white. All right? And all in all marks lapho or 6 marks. Don't forget I'm no P2 phezulu. F2 ngenzansi, okay? And also I've got meiosis nobani no fertilization. So now I know we call our 4 hanging marks, okay? 4 low hanging marks. Eyokuqala ukubhala iphenotype osukunezo yena. Okwesibili genotype usukuneziwe futhi naku question or ku genetic problem. Elesithathu ukubhala meiosis no fertilization. Elokugcina ukubhala P1 no F or P2 no F2. And it gives you 4 marks. 4 free marks. 4 low hanging marks. Okay? And remember, a genetic problem is out of 6. So I hope with my very genetic problem go examination question paper. So it told us all. Alright. Then let's move on now to the last type of dominance. And that type of dominance is called codominance. Codominance. So lana ke lakhona futhi alikho igene elidominant kunelinye. So kuma offsprings noma einganeni avela womabili. So isibone ukuthi kunenkomo enombala omhlophe nomlabala omnyama. So lapho sokuhlangana inkomo enesikhumba esimhlophe nenkomo enesikhumba esimnyama. Womabili ayavela lapha einganeni. Sekuvela ingane enamabala amhlophe namabala amnyama. So masichaza kahle ke ngesithi yini ngempela na kuthiwa it refers to a genetic cross in which both alleles are equally expressed in the phenotype. So ihluke ngani na ku incomplete dominance. Incomplete dominance kuvela into entsha engafani nabazali. Sabona bona kanti lana kukhona ukubona ukuthi lokho kababa lokho ka mama. So ngabe amazinjo besho bathini lapha bathi both alleles are equally expressed. So avela ngulingana. All right? In the F1 generation or F2 generation. All right? Akasibheke ilakwa I example yakhona. noma isibonelo segenetic problem now eveza a principle of co-dominance. All right, but a plant with white flowers was cross-pollinated with a plant with red flowers. All the plants that grow from the cross had flowers which are equal distribution of red and white color. So now there's no gene which masks the other gene. Both genes or characteristics are shown on the offsprings. Ziyavela zombili lapha ingane. Nanta iflower lapho na o red nobani no white uyavela. All right. Then now represent this genetic cross to show the possible genotypes and phenotypes of the F1 generation of plants. All right. Now a red flower was cross-pollinated with a white flower. P1, the phenotype, red crossed with white. 2, the genotype. Both of these plants were homozygous. So capital R ababili for red and not capital letter W about 2 for white. Remember, incomplete dominance and co-dominance, we don't use small letter, okay? We use only the capital letters because both these genes are displayed on the banner of the offsprings. All right? Third step, meiosis. Red will produce. Red is going to produce gametes aga red. White is going to produce gametes ga white. All right? And then basically fertilization. Lapho ke sekuzovela manje ke zonke ingane zovela zino red and white. Bobabili bayavela kuphina enganeni. That is incomplete dominance. So those are the 3 types of dominance. Those are the 3 types of dominance. Incomplete dominance, lapho sibona khona kuvela i-characteristic A1, i-trait A1 noma uvela ufuzo olulodwa. Okwesibili, i-incomplete dominance, lapho siyabona ukuthi alikho igene elihlula elinye. All right? And ingane ezivelayo azifani nabazali. Abasho ukuthi ke mohlanisa iflower elibomvu neflower elimhlophe kuvela ingane ezipinki ebona ayifana abazali lokho sithi incomplete dominance noma intermediate dominance. All right, sindlule lapho siyobheka manje i-co-dominance. Ku-co-dominance kuvela kokubili, both ama-parents ayavela lapho ku-co-dominance. All right, sindlule lapho ke sesiboleka ukuthi sakhuluma inheritance of just ama-characteristic and we're looking at only one characteristic, i-monohybrid cross. Sibuye lapho manje ukuthi inheritance of sex lesibiza ukuthi ke is sex determination. All right, so when we talk of a sex determination sisibheka ama-chromosomes. Sibuye lapha khumbula ukuthi thina bantu we've got 46 chromosomes, and ama-chromosomes they always occur in pairs ahamba ngamabili. Ukuthi sinama-pair amangakina awu-23. We've got 23 pairs of chromosomes. All right, so out of these 23 pairs, 22 pairs noma awu-44 which are responsible for the formation of body cells or somatic cells, ama-cells omzimba, ikhanda, indlebe, inyawo, bonke, all the different parts of the body are formed by somatic cells, ama-somatic cells. Bese kuba khona nje amabili ama-chromosomes lawo aseveza ubulili ukuthi ngabe umfana noma intombazana, and lokho sibiza ukuthi ke i-sex determination. Sex determination. So ngamanye amazwi, out of 46 chromosomes, 44 chromosomes kubantu besifazane are autosomes. We call them the autosomes. They are responsible for body cells, somatic cells. Bese ukuthi amabili siwabiza ukuthi ama-gonosomes, ukuthi forma ina ama-sex cells. Kumuntu wesifazane kuba 44 plus XX because 2 chromosomes. Kanti kubaba noma kumale nomuntu wesilisa kuba 44 plus XY. So umama uXX, ubaba uXY. Umfana XXXY, intombazane uXX. All right. Let's do a genetic problem now. Which sibathola kanjani abantwana abafana nabantwana bamantombazane? But a couple has got 3 sons and a woman is pregnant again. Show diagrammatically by means of a genetic cross what percentage chance of a couple of having a baby who is a girl. Now, basebenzela abantu banabafana bathathu. Usekhulelwe umama manje ingane yesi4. Sekuthiwa kubuzithiwa ke ngabe anganani ama chances? What percentage ukuthi bathole ingane yani yentombazane? Sebenze 3 zonke ingane. Lesi4 ngaba yini? Ngabafana amantombazane. Asibe ukuthi ama chances ukuthi ke kube intombazane angakanani. Same thing futhi. Njengoba siqale siqale nje monohybrid cross. Nalapho we've got P1, a female, a XX, and a male, a XY. We cross XX no banana XY. So for a phenotype, you write male crossed with a female. Step 1, maglacolate. Genotype, XY for male crossed with XX for female. Then the meiosis. So email is okippa or X must be Y, no banana, no Y. But if female yona ekippa or X board. Alright? So mawufaka lapha uthatha uX ndoka baba umhlanza no X kamama kuphuma intombazane. Mawuthatha uY kababa umhlanza nomunye X kamama kuphuma ini umfana. So ngamazi ithini phenotypic ratio na 1:1. Okay? Okusho ukuba na u50% uthola umfana, u50% uthola intombazane. So the correct answer there is 50% chance. Ngoba sonke isikhathi noma nje abantu bafana bakaba u8 kwale mayiphinde khulele umama amathu ukuthi uthola umfana ingane yomfana nentombazane ayahlala enjana elingana. Uma ubheka lapha ke umuntu ngempela ozithi la ekhaya kube labafana ubani ngabe ubaba noma umama uthini namfundi ukhumbule njalo ukuthi ubaba uno XY so ubaba kuphela okhipha oY so ubaba mayikhipha oX bodwa kuzoba amantombazane wodwa ubaba mayikhipha oY bodwa kuzoba abafana bodwa kodwa mayikhipha oX nabo Y ama chances okuthola abafana namantombazane ayalingana all the time and lo sikubiza ukuthi ke is sex determination. All right, bese kuba khona ke lolo fuzo lolo oluhambelana nobulili. All right, like for instance siyazi abantu besilisa abafana nesifazane. All right, and also kunezifo othothi ziphatha abantu besilisa kuphela ezingabaphathi abesifazane khona iphatha abesifazane ezingabaphathi abesilisa. So lana ke la grade 12 sizobale sobheka ama sex linked characteristics amabili and usually aphatha abantu besilisa and we say they are called sex linked disorder. They affect males more than females. We'll start with the first one. Sex linked characteristics we bheka lapha hemophilia. Iyini hemophilia na? It is a disease where the blood fails to clot. It is ukuthi khona abantu abalesifo sokopha. Uma elimala umuntu wakhona alikhandeki ihluli. All right? Wopha nje uwaya kuyoze kuyovala. So lo muntu onjalo ke ufanele maqala nje limala aphuthumise isibhedlela umuntu esizoqala then umphuthumise isibhedlela because alivaleki. Yini blood clot ikhandeka kanjani na? Whenever you cut your skin mawusike kanje nomulimala phuma igazi leli gazi limalishaya nomoya kuneprotein fibrinogen lapha it mayishaya ihlangane neoxygen bese ikhanda ifibrin bese ikhanda ama fibers fiber protein a network of fibers kuleyo ndawo esiyekile so mase kudlula igazi lapha ama blood platelets afika ablockeke lapha bese ikhanda ihlule legazi and ingcaba bese yavaleka so umuntu onehemophilia ayenzeki le nto leyo failure of blood clotting. Mage laphuma igazi liphula waya alikhandeki ihlule ukuze avaleke lo. All right. So now, but hemophilia is a sex-linked hereditary disease that occurs as a result of a recessive allele, which is only found on the X chromosomes. So Mawibala, you write X low, chromosomes, then you put a capital letter H peso. Low should be normal, low muntu low. All right? And itholakali Y chromosome. So a normal father and heterozygous normal mother have children. Represent a genetic cross to determine the possible genotypes and the genotypes of their children. All right? And now, followers, the alleles for hemophilia are indicated as a superscripts of sex chromosomes. For instance, in a female, a normal, it will be XX, but phezulu superscript kubhalwa H capital letter. All right? In a normal male, it will be XY, base ubhala X capital letter X. And remember, there is no gene in a Y chromosome. Y chromosome alikho le gene altogether. Litholakala kuphi ku X chromosome. So uma umfana normal uba no XY bese kuthi lapha ku X ufaka capital letter H. Kodwa ufana mayigula ephethwe hemophilia ubhala XY bese ufaka small letter H lapha phezulu kuba ku X. All right? I think you understand that one. All right. Ake sibheke manje solution lapha. Uthiwa lapha a normal father and a heterozygous mother. So umama heterozygous akaguli, but she carries that gene. She is a carrier. All right, let's write the genetic problem. Step 1, P1, first parents. Heterozygous father, heterozygous mother, or a carrier, which is normal mother, or normal mother, okay? Is crossed with a normal father. Good genotype. Umama will be XX. One of the X's will have a capital letter H, and the other will have a small letter H, because umama is heterozygous. And then it's crossed with a normal father, which means ubaba no X, H, capital letter, no Y. Remember, this gene is not found on the Y chromosome. Sekwenzeka in manje meiosis sekhandeka in ama-gametes. So ama-sperm kababa amanye azoba no X ophilayo no Y. Umama amaqanda akhe azoba no X ophilayo no X okhulayo. And mase bayacrosswa lapha. Mase crossa during fertilization, remember? So u-X lo kababa onormal ehlangana no X kamama onormal kuvela ingane yentombazane ephilayo noma enormal. But X kababa lo normal ahlangana no X kamama lo ogulayo kuvela futhi ingane yentombazane ephilayo, but lena is the carrier. All right? Size ku Y. U-Y kababa ahlangana no X kamama lo ophilayo X kuphuma umfana onormal. Bese uthi ma u-Y kababa ayesehlangana no X manje lo kamama ogulayo sekuvela umfana ogulayo. Uyabona sigula yini amantombazane amathathu lapha. Wonke ayaphila, qha umfana useyagula. And remember, ubaba akagula, umama akagula. Naku umbuzo engizokubuza wona ke. Ubani ogulisa ingane la ekhaya? Sengushiya nawe lo umbuzo ke loyo. All right. Sibuye masi analyze kahle ke lento lena. Ukuthi ke into eyenza ukuthi iphathe abantu besilisa isikhathi esiningi. It is because abantu besilisa abanalo elinye igene elizokwazi ukumaska the gene error recessive. This one is caused by a recessive gene which is found on an X chromosome. So that's why it affects males than females. Affect males more than females. Why does hemophilia affect males more than females? The answer is one, it is because the Y chromosome is smaller and does not have a gene to mask this recessive gene from an X chromosome. So whenever the male receives a gene from the mother, which is recessive, or the gene for him from mother, definitely sure that male is going to be hemophilic. And the second type of the sex-linked inheritance, we follow the same procedure. Color blindness. Now, color blindness, it affects males more than the females. So males nama females because of the affector layer. This chromosome is also found on the X chromosomes. But with color blindness, we don't use letter H, but we use letter B. Capital letter B for normal and small letter b for abnormal color blindness or affected male and females. All right. So we're going to go inheritance of the sex-linked characteristics. All right. I guess basically just two problems. All right. We look at the problems amabili on monohybrid cross involving the different types of dominance as well as sex determination. Problem number one. Fair color in mice is controlled by a gene with two alleles. A homozygous mouse with black fur was crossed with a homozygous mouse with brown fur. You start writing down. They are telling you that the fair color of mice is controlled by a gene with two alleles. I was doing it. All right? But with a homozygous mouse with black fur, homozygous black. So I'm was crossed with a homozygous mouse with brown. So we're going to write black and brown, and they're both homozygous, which means that the alleles are the same in the locus. All the offsprings now had black fur. So we actually which one is dominant and which one is recessive. And no black fur. Principle of dominance. There is a principle of dominance. So black is dominant over brown. So you want to analyze the problem before you go to the question. You read the sentence, you write something down. No problem is this card. So we are part of black is dominant, capital letter P, then small letter P for brown. Alright? Using symbols capital letter P and small letter P to represent the two alleles for fair color, show diagrammatically a genetic cross between the mouse that is heterozygous for color and the mouse that is brown fur. Show the possible genotypes and phenotypes of their offsprings. All right. I'm going to give you a few moments. We the problem. Your pen and paper is ready. All right, Bafundi. And tell what he has in open legal muzzle. I guess very impeded what eating it up. All right. So, Sakuba Ninja Supala P1, phenotype, Besa crosser O black, no brown. All right. Heterozygous black and homozygous brown. Yes. So let's win each genotype. Capital letter P for O black, small letter P. For O brown, or small letter P, ababil. What's the meiosis? So now black laws will keep amagamis au capital letter P, amanya abel capital letter, small letter P. Or to do P for black, or small letter P for brown. Those long or homozygous brown. Also faka or small letter P ababil. Base we are cross eye. So u black, no brown, kupuma heterozygous black. Pena black, no money brown foot or money heterozygous black. About two, And then bese kuthi brown no brown ngale kuphuma ingane njani na brown, nomunye brown futhi no brown ngale ku female kuphuma brown. So ngamanye amazwi 2 brown and 2 black phenotype leyo, and the percentage lapho kuba na 50-50%. So ezimbili ingane zizoba black, ezimbili zibe brown. All right, so awufakele kwemqondweni ke sonke isikhathi uma ucrossa uheterozygous neheterozygous, you get a ratio of 3:1. There will be 3 black and 1 brown. Kanti uma ucrossa heterozygous ne-homozygous, i-ratio will be 50-50. Okay, okusho ukuthi ithini ratio na? Phenotypic ratio 2:2 noma 1:1, ayalingana ama-chances. Niyabona yilo kade sikwenza lapha ngikhathi sikhuluma nge-sex determination, umfana nentombazane, ukuthi amathuba okuzala ingane yomfana ayalingana namathuba okuzala intombazane. All right, bese buka enye futhi genetic problem yesibili. In rabbits, a dominant allele, capital letter B, produce a black fur. All right, and there's a black fur, and a white fur, small letter B, produces a white fur. All right, this is the allele, the small letter B. Use genetic cross to show the possible genotypes and phenotypes of the F1 generation for a fair color if 2 heterozygous rabbits are crossed. So they are heterozygous. All right. Most biology problem is very simple. Cross a heterozygous nani ne heterozygous. Any ratio for you to the lab, it's correct. For 3:1. 3 black and 1 white. As in the name. P1. So lab we write the phenotype. Black, some cross and no black. It's 1 mark. That's a mark for that one. The genotype, heterozygous black, no heterozygous black. So lapa, capital letter B and small letter B. Nangale futi, capital letter B and small letter B. Then we move on to meiosis. So each of one of them, the black one, is going to produce we male ama sperm anobana ano black, amanye azobanobana no white. But ngapha female same thing. Amaqanda amanye azoba no black, ali amaqanda aphethe black, amachinza khona, amanye aphethe white. Then masu across all. Mokrosa lo black, nalo black we female, phuma a homozygous black offspring who F1 generation yakho. Bese kuthi esibili, when you cross this black and white, phuma heterozygous black. Okay? But if you cross black white, no black wangale phuma heterozygous black. Base cross white ngapha no white ngale phuma na homozygous white. Seliyavela la gene la bana la white, even though abazali bobabili bemnyama sebethola ingane emhlophe. And that is the principle of segregation. And the ratio lapha nje bathi ngishona seqaleni, there will be 3 blacks and 1 white. That is the phenotypic ratio. And each genotypic ratio itina, there will be 1 homozygous black, 2 heterozygous black, and 1 homozygous white. Ake bantu bami ke sesifike esiphethweni sesifundo sethu namhlanje. Engithemba ukuthi abafundi balelisile bezwa, bese ngicela ukuthi ke uzothatha incwadi ezinye, understanding life sciences. Also you can take in mind the gap and answer the questions at the end of this subtopic. And also ubheke amaphepha eminyaka edlule and uwasolve onke ama-genetic problems, yonke amaphepha eminyaka edlule, as well as amaphepha from other provinces in preparation for the exams. Remember, bantu bami, you read a question carefully before you answer it. And lapho life sciences, you are given the diagrams. You study the diagram below and answer the question that follow. Before you look at the question, you analyze the diagram. All right? Uma kufanele uyilabel, you label the diagram. You put all the information, everything that you know about that particular diagram before you start answering the questions. Log is going to save you time, because when you come to questions, sekubalula ukuthi ke uvele ubhale ini ama-answers ngoba usuwabhalile noma ngepeni nomsizi lapho ku-question paper yakho. All right, bye. Imo wenu, PS Mabaso. Bye.
Siyanibonga ngokufunda nathi. Siyanimema ukuthi nithamele futhi nakusasa. Ningazithola lezi zifundo ekhasini lethu lika Facebook elithi Go Digital SA Foundation na ku YouTube elithi Go Digital SA Foundation. Ziyatholakala na ku Khosi FM njalo ngoLwesine kusukela ngo9 kuya ku10 ebusuku. Ningasithola nango WhatsApp kwinamba yethu ethi 068 151 107.